your question about the microbiome. So, we've heard a lot about the brain. We've heard a lot at these different levels, and we're moving from human cells in some ways to bacteria, and the microbiome is the topic of our next presenter focus. So we go from a computational biologist to a PChem guy who's worked and triumphed as a kind of a microbiome champion, looking at dysregulation of the microbiome. In the real world, we think about that a lot, right, with kind of what's going in from our mouth all the way through, it lives on our skin and our gut. Moms think about it a lot and try to avoid C-sections because that pass in the beautiful moment of birth for a child, they come through the vaginal canal, they're covered in bacteria, and that bacteria can be life-changing. We know a single dose of antibiotics in an infant's life can potentially regulate differently their likelihood of development of things like Crohn's and IBD. And we have all sorts of, antibiotic-resistant infections. And in fact, last year, 5 million human beings on this planet died because of antimicrobial-resistant organisms. So, Dr. Nebojsa Janjic is coming up. He's going to talk to us about this incredible microbiome and all of the things he knows. Thank you.
Thank you, Wendy, for that great introduction. In fact, you said a number of things that are really important. Thank you, Larry and Larry, for an invitation to come here. It is great to see so many familiar faces here. As some of you know, I've been in Boulder for quite a few years, and it's been a privilege to really work in this town. I am going to talk about bacteria. And as we do that, it's useful to think about how bacteria fit into the grand scheme of life and these trees of life. And so, are they plants? Are they animals? Darwin pretty much dealt with these two groups. But his contemporary, Ernst Haeckel, did carve out in this middle tree a category for basically the microbes, and he called them protista. When you look at bacteria, which were at the time called moniers or monera, they're usually put at the very bottom of the tree, almost at the root, in several versions of the tree that he came up with predictably man being near the top. So we now know that all life has similar molecular machinery, and both Mike and Art have introduced this concept of central dogma of biology, where DNA makes RNA, makes protein. And so we can now pick a gene that is shared among all living organisms, like ribosomal RNA. We can look at the sequences, we can read them, and simply put sequences that are similar close to each other, and those that are further apart in the tree. And when we do that, we see a tree of life that's quite a bit different. It does have archaea, which were originally called archaebacteria, then bacteria, and then eukarya that contain plants and animals. So one of the consequences of this development is that we can now detect organisms that we can't grow in culture, which is the vast majority of organisms. And our own Norm Pace has done quite a bit of this work. He very much liked to be out there in the field and would find rich bacterial communities in places like thermal vents and caves. And then some places where you would hope there wouldn't be all that many bacteria, like New York subways and shower curtains and such places. But there are clearly also a lot of bacteria in our own intestines. So I now want to add a component of time. This is a massively abridged history of the Earth, showing only the events that I want to show you. So we begin with moon-forming impact. This is a time when a Mars-sized object hit the Earth. And so for the purpose of life on Earth at that time, this would have effectively sterilized the planet. So we kind of reset everything at that time. And then first life emerged sometime after that. Then a bunch of time goes by and there's this great oxygenation event. And then a whole lot more time goes on, and there are such things like Cambrian explosion and dinosaurs and mammals and finally humans. So this is a much more refined version of all of this that was recently published in Science. And the key point to keep in mind is that before this great oxygenation event, where oxygen emerged on the planet, anaerobic organisms was all there was on the planet. And so those are shown in the blue bars in this dendrogram, and red is aerobic organisms. So you can see that anaerobic organisms preceded aerobic ones by quite a long time. So the organisms that were anaerobic when oxygen emerged had basically two options: adapt or find ecological holes where oxygen is absent. And so it turns out that the intestines of animals are anaerobic environments, and consequently more than 90% of our gut bacteria are obligate anaerobes. Now, we of course think of ourselves as hosts to bacteria, and bacteria, which by the way in my presentation will be able to speak and have an opinion about things from time to time, they may think that there's a question about who is the host and who is the guest here. When we think about the number of cells we have in our body compared to the bacterial cells we have, things are pretty much on par, and bacterial cells are referred to as microbiota. When we look at the genes, and there are about 20,000 genes in the human genome, and because there's the same copy of DNA in every human cell, and there are many species of bacteria that we host, there's at least 100 to 1,000 more genes that bacteria have. So, we can wonder again about who is the host, who is the guest. The good thing is that many gut bacteria are beneficial, so they do useful things, as Wendy mentioned. They help us extract food or energy from food. They make things that feed our intestinal epithelial cells, like short-chain fatty acids. They stimulate our immune system. And quite importantly, they protect us collectively from pathogens in what's known as colonization resistance. So modern life, again, as Wendy noted, has in fact compromised our microbial diversity. We have in fact become a bit of germophobes. We eat too much processed food, too little fiber. We definitely use too many antibiotics. The family size is smaller. There's less breastfeeding and more C-section. So all of these things combined can contribute to reduced microbial diversity that we have. When we weaken our colonization resistance, and again, this is a term that means this is what protects us from pathogens, problems can arise. Most of the infections that we get are treated with broad-spectrum antibiotics, and the reason for this is because most doctors don't have the right diagnostic tools, because they actually don't exist, to rapidly identify which pathogen is causing the infection. So we come in with these huge guns and we basically wipe out the infection, and along that there's a collateral damage to the intestinal flora. There is a condition called antibiotic-associated diarrhea, which was not known to have a cause specifically, but now we know that it's caused by a specific organism called Clostridioides difficile, which I'm going to represent as this spar or a rod. So we begin with a nice, rich intestinal flora. There may well be some C. diff around, and then we get treated with an antibiotic, which causes massive dysbiosis. And because C. diff is an opportunistic, fast-growing bug, it can quickly overtake the space that's available and become the dominant organism in the gut. The reason this is a problem because C. diff strains that are able to make toxins are clearly bad bugs. These toxins are encoded on what's called pathogenicity island, and they have receptors on intestinal epithelial cells that allow them to get in. When they get in, they modify through a process known as glucosylation a family of protein called Rho GTPases, which are then inhibited, and that disrupts the cellular cytoskeleton. It leads to breakdown in tight junctions between epithelial cells, cell rounding, and finally cell death. C. diff also produces metabolites that are made from some amino acids like tyrosine. And the example I want to show you is a metabolite called paracresol, which can be found in millimolar level in stool. And I want to be careful to include molar concentrations of things for Larry's benefit because I know that he will want to know that. And paracresol is definitely used as a biological warfare agent that C. diff uses against other bacteria. So it actively suppresses other bacteria to create more room for itself and therefore has a growth advantage. And we'll come back to paracresol later. So some nurses claim they can actually walk into a patient's room and smell C. diff on patients. I see Katie nodding her head. So this was put to some scrutiny, perhaps in an unfair way, where the nurses were only allowed to do the smell test without ever seeing a patient. And so perhaps this is not a great standalone diagnostic test. Maybe it's a rule-out test. But what there isn't any question about is that C. diff patients had definitely higher levels of C. diff, which is a volatile organic compound as it's known. So not all antibiotics are the same in terms of their propensity to cause C. diff. Some are actually kind of okay, like linezolid and some of the tetracyclines. But essentially, the broader the spectrum, the worse the problem is. Some of the very worst antibiotics are these super broad spectrum beta-lactam antibiotics like carbapenems, as well as fluoroquinolone antibiotics. One other trick that C. diff has is it can form spores. So it can go from a nice vegetative cell that looks like a rod, and under stress, and they can be the dehydration or exposure to alcohol or antibiotics or lack of nutrients, it can form spores. Spores are basically a dried up form of the organism, and they can persist in the environment for a very long time. Now, when good times come back, spores can germinate and become vegetative cells. So this is kind of an evolutionary superpower. I mean, if you think about it for a moment, you know, if you can make spores, you don't need to be nice to your host, and you don't need to be nice to your neighbors. They're very easy to spread. They can survive in harsh conditions. And so your life cycle is basically you get into a host, you hit, you run. In the case of C. diff, yes, that way. And then you can sleep for a very long time, and you can travel near and far. And when conditions are again ripe, which means you might be ingested by an animal, you repeat the cycle. And so this is why these things are really difficult to get rid of, which is related to the problem of C. diff infection, which remains, according to CDC, an urgent problem. So about 500,000 cases a year, and very high death rate, and these C. diff spores are in fact widely spread. And so this is an imprint of a sterile glove on a petri dish, which illustrates the problem. The biggest clinical problem with C. diff infection is not so much our ability to clear the infection once we detect it. There are a few antibiotics that can do that, beginning with metronidazole, which is a horrendous treatment option for C. diff, which is now discouraged. But then even the vancomycin and the latest antibiotic, fidaxomicin. The biggest problem is recurrence. After the first episode of C. diff, recurrence rates are in the 25% range. But now when you cure and you recur, the recurrence rate from that stage jumps to 45%. And then if that happens again, 65%. And you can imagine that the patient's condition goes quickly downhill when that happens. So this is why it's so bad. So the reason recurrence is a problem is because taking vancomycin, which is by far the most commonly used antibiotic for C. diff infection, something like 80% of C. diff cases are treated with it, is also a broad spectrum antibiotic. So you might begin with a lot of C. diff, which you might remember are these blue rods. You can get rid of most of them, but you're left in a state of dysbiosis, which got you in trouble in the first place. And what happens is you get recurrence. So an optimal treatment would be, again, we come back to the central dogma. What you would like to do is stop proteins from being made, which would mean you would stop toxins from being made, but you also stop spores from being made because new spores need to make spore coat proteins and other things to make spores, which is not possible if you stop protein synthesis. So we have been pursuing an enzyme called methionine tRNA synthetase, or METRS, which is a novel drug target, which is an enzyme that loads amino acid methionine onto tRNA. tRNA is this squiggly line which is made of RNA, and it's kind of a magical adapter molecule that converts a four-letter code of DNA and RNA into the 20-letter code of amino acids. And it does that by recognizing these triplets that are codons, and then on the other end of tRNA are the amino acids. And so METRS loads amino acid methionine onto the tRNA, and if you can stop that, you can stop protein synthesis. METRS is a bit unique in that it inhibits loading of the first amino acid in protein sequence of bacteria, which is formyl methionine. And so this will inhibit the initiation stage, but it also prevents the ordinary methionine from being loaded, which is a part of the elongation state. And the difference between eukaryotic and bacterial enzymes is quite large. So we have identified a molecule. It's a small molecule that inhibits this enzyme quite potently. We originally brought a series from GSK, and then we optimized the molecule for C. diff. And so now we call this molecule CRS3123, which does have one chiral center, as chemists would call it, which is right there. And it has really good potency of about 20 picomolar, but only with the right enantiomer. And it has good microbiological activity as measured by this minimal inhibitory concentration of 0.5 versus way more than 32 for the other enantiomer. So we do understand the mechanism of action of this enzyme, which sits at the active site where methionine would sit. And so it's a competitive inhibitor of methionine binding. But importantly, it binds in a cooperative manner with a second substrate, which is ATP. And this is important because there's a lot of ATP inside the cell, so you would rather not have to compete with it for binding. And that results in the high potency that we have seen. Now, narrow spectrum, which we're seeking, can be explained in terms of the phylogeny of now METRS enzymes. So this is phylogeny at the level of amino acid sequence. So it turns out there are two types of this enzyme, type 1 and type 2. Type 1 is at the top and is present in C. diff, as well as a number of other pathogenic bacteria. But the vast majority of gram-negative bacteria, as well as most of the intestinal anaerobes, have a type 2 enzyme against which CRS-3123 is not active. I'm going to now jump over a huge amount of preclinical development to tell you that we have been able to advance this molecule into phase 1 clinical trials. We have done all of this with NIAID, and we have done 2 such studies. In the first one, we have given CRS-3123 once in doses going up to 1.2 grams, and then repeated that in a 10-day treatment course, given this drug candidate twice a day for 10 days at 3 different doses, again going up to 1.2 grams. What we have seen is very low oral bioavailability. Now, this is actually a really good thing for C. diff infection because that's where C. diff is. It's in the gut. So we're seeing high levels of this drug candidate in the gut, which are way above the MIC, which I've shown you before, which is 1. It's also above what this band called mutant prevention concentration. I'm going to try to stay within my lines here. And so that should attenuate the emergence of resistance. We have also seen relatively minimal to moderate effect on normal gut flora, as we hope to see because of the narrow spectrum. In this chart, what you're looking at is all of the bacteria in the gut lumped into major categories called phyla. And so if you look at the effect of the placebo, and this is over time on the x-axis, during the 10-day treatment, the placebo, and the 2 major categories are firmicutes and then Bacteroides. And so you can see not a whole lot going on during the treatment. The same is pretty much true at the 200 milligram dose, less so at 400 and even less at 600. But importantly, no phyla are lost. And we did all this by this small subunit 16S ribosomal RNA sequence. So this informed us, along with the previous experiment about the concentration in the gut. Again, we want high levels to go into phase 2. Design of our phase 2 study was pretty conventional, a 10-day treatment course, assessment of whether there's a cure or not by day 12. And then we did follow these patients up, as the FDA encourages sponsors to do, through day 40, but also we went later to day 70. And then the primary and secondary endpoints are cure at test of cure, day 12, as well as looking at day 40 to see what happens. So what we have seen are high initial cure rates, comparable to that of the vancomycin, well above 90% for both of the dose groups of CRS-3123. But the difference really emerged when we looked at the recurrence. Recurrence rate with vancomycin was in a typical 23% range, whereas we only saw 4% with CRS-3123. So, and we also saw that the molecule was safe and well tolerated in that the rates of adverse events were comparable across these groups. So I could stop the presentation here. This is conventional drug development. Wendy would be cheering because I would be way ahead of schedule. But I'm going to tell you a few other things and ideas that have surfaced since, and I do believe I have a few minutes. I'm going to talk about autism. As many of you know, the rates of autism have been increasing for reasons that are really not clear. It does affect boys 4 times more than girls. It affects everybody. There is definitely a genetic component. There are more than 100 genes that contribute to autism. So sorting this out is not trivial. There are no FDA-approved treatments for core symptoms, and you don't really need to cure anybody. You're probably not going to cure anybody, but if you can improve behavior, even to some degree, you would do a world of good. So it turns out that GI problems are quite common in autism. They're about 3 times more frequent compared to neurotypical individuals. This is constipation, diarrhea, bloating, pain. And the GI symptoms are actually strongly correlated with autism symptoms. Gut microbiome is less diverse in autistic individuals, as is GI motility is reduced, and leaky gut, as it's called, is also more common. So at this point, I want to introduce you to Ellen Bolte. So her son Andrew was developing normally until he was just about 2 years of age. He got an ear infection for which he was treated with multiple courses of broad-spectrum antibiotics. He developed diarrhea. Now, all of these things so far are super common, and then he rapidly descended into severe autism. Late onset or regressive autism occurs in about 1/3 of the cases. There tends to be a history of antibiotic use. Onset of symptoms often follows closely antimicrobial therapy, and GI symptoms are common and often persist. So it turns out that dysbiosis in autism does follow directional trends. And this is the most efficient way I could summarize a whole bunch of literature that comes from a review, but basically there are 2 main trends. There is an increase in Clostridium cluster, or Clostridioides cluster as it's now called. Most of these organisms are spore formers, and they do produce enterotoxins, neurotoxins, as well as toxic metabolites. There's concurrently a decrease in widely acknowledged beneficial bacteria that are in the bifidobacterium cluster. So I want to point out to 2 organisms, Clostridium perfringens, and then Clostridium boltiae, which, yes, was named after Ellen Bolte. So at this point, what Ellen did is she looked for microbiologists who really understood intestinal microbiome, and she found Sid Feingold at UCLA. Sid was a scientist who published something like 500 papers in this field, so everybody knew him. So she convinced him to test oral vancomycin against Andrew, who improved. Then they expanded this to a small phase 2 study where they enrolled kids who had this sequence of events. Some kind of an infection, treatment with antibiotic, diarrhea, regression into autism, and 8 out of 10 of them improved. But the effect didn't last. And so in 2010, Sid called us. And the reason he called us is because he saw a paper that we had published on the activity of CRS3-123, and what he noticed, again, these values are MIC, minimal inhibitory concentration values. A value of 1 is widely acknowledged to be kind of a potent antibiotic. Less than that is even more potent, and then above that, questionable. So what he saw is that compared to vancomycin, we had activity against all of the suspected contributor to autism symptoms, but was much cleaner in terms of spectrum. So I'm now going to briefly talk also about paracresol, which I mentioned before for C. diff. It turns out that in 17 studies where this was examined, all reported higher levels of paracresol in autistic individuals. Now, is this just, and levels correlate with symptoms, so is this just correlation? I mean, that should always be a question. So this experiment got done a few years ago where a group in France put paracresol in the drinking water of mice, or gave them just water. The ones who drank paracresol 2 millimolar became less social. The ones who drank normal water were hanging out with other mice, which mice like to do. When you transplant, when you do a fecal microbiota transplant from normal mice into mice that were behaving in an autistic manner, they actually reverted to typical behavior, and vice versa. And this effect is not subtle. So this is as close as I have to actually causation. So paracresol is a well-known metabolite that's widely perceived to be not good. It affects a number of different organs, including the brain. So when you look at the bacteria in the gut that are able to make paracresol, there are at least 55 different ones that can make it, but they're not all alike in that it's clearly a bimodal distribution where many of them make some, but not all that much. But 4 make way more than others, and their names are listed here. And so we tested our drug candidate against all of them, and it turns out it's active because all have METRS1 and not METRS2. So coming back to vancomycin and its short effect, Jim Adams and his colleague Rosie Kreimlich-Brown at Arizona State University decided to follow up on the vancomycin study that Sid Feingold and Ellen Bolte reported. And they began with the vancomycin treatment. Then they did a bowel cleanse of a type that many of us have done ahead of a colonoscopy. And then they transplanted the fecal microbiota from neurotypical individuals. And when they did that, they saw improvement in both GI symptoms as well as autism symptoms. But this time, the effect persisted for at least 2 years. And so on the y-axis is this childhood autism rating scale. It's one of the most widely used scales. And these bands in pink will show you severe, mild to moderate to basically very little. And you can see that over this period of time, some kids actually ended up in the neurotypical range. So pretty exciting observation. So we are wondering whether CRS-3123, for reasons that I mentioned in terms of its spectrum, might have a similar but lasting effect. And so we have gone so far as to, because we already have an IND for C. diff infection, we filed a separate IND for autism, for which the FDA has now given us a study may proceed letter. We are going to look for whether there is an improvement in GI symptoms in individuals who have them at the study entry. We are going to enroll people who have elevated levels of these suspected Clostridia, and we are very much considering now to also add another inclusion criteria that would mean elevated levels of paracresol. And then the question is just what might be the effect size, if any, for both behavior and GI symptoms, and does, importantly, narrow spectrum translate to less dysbiosis, therefore longer lasting effect. And I'm going to conclude with an autism-like rare disease, again knowing that Larry is interested in rare disease, and in fact many of you as well. Wendy, I know you are. So these individuals are exhibit autism-like behavioral symptoms. They have very unique facial features. This was initially described in 1978. It's a mutation in a single gene, transcription factor 4 on chromosome 18. At least 70% of these individuals have GI dysfunction, and this is a huge problem for them as it is for their families. So it turns out that they have elevated levels of one of these Clostridia that we are suspecting contribute to symptoms. So we are going to be thinking about this as well. So now I will wrap up by just saying that the target phylogeny of METRS enzyme explains not just the narrow spectrum, but some of the indications we're pursuing. C. diff infection obviously targets C. diff. For autism and Pitt-Hopkins, these are these two other Clostridia. And then we're also thinking about graft versus host disease, because it turns out that if you have elevated levels of some of the enterococci, you have, after bone marrow transplantation, you have a much higher risk of graft versus host disease for reasons that are really not understood. So the idea would be just prophylactic treatment ahead of bone marrow transplant. And so with that, I want to end with Dunning-Kruger effect and when do we know enough to do something? So, we begin by learning just a little bit and we're thinking this is great. And so Mark Twain had an opinion about this and in his Southern way described kind of not this affliction. And so then we learned stuff and, things are harder than we thought to extreme frustration. And then we begin to kind of get a hang of it and think that this is cool but complicated. So we are somewhere on this curve, but very excited about what may come. So with that, I want to thank a very large number of colleagues who have contributed to this over the years, especially my co-founders at Crestone, Urs Oxner, Thale Jarvis, and John Roloff. We owe an enormous debt of gratitude to our NIAID colleagues who have supported all of the clinical development so far. Kathy Lozapponi for help with microbiology at the University of Colorado. And then many of my colleagues who were at Replodyne before, and especially Jim Adams and Rosie Cry-Malik-Brown at Arizona State University for their input into our autism study. So with that, I want to thank you. I'll be glad to take questions.
Well, that was amazing. Thank you so much. You're such an incredible teacher. That one didn't feel quite so over my head as a pediatrician, but I'll tell you, at a time when autism is right in the front of everyone's minds because you can't avoid it, I think these kind of discoveries with such fastidiousness and such excitement really can bring us a sense of hope. So with that, as many questions as we can squeeze in, we are going to take a break after this. We have about 10 minutes for questions, and then we will reconvene at 4 in about a half an hour. So you can always find them at the break. You go first.
That was incredibly fascinating. So as you know, autism has very, very high heritability. And so there's lots of questions that come out of that. And one of them is, does dysbiosis also have high heritability? And apart from that one gene, TCF4,
Yes.
that you mentioned,
Yeah.
there have been so many other loci identified for autism. Are they also involved in dysbiosis?
That is an area that is not well studied at the moment. I, in fact, just attended an autism conference in Seattle 2 weeks ago. And there was a description of one of the main genetic drivers of autism. Again, mutation in a single gene. The percent of GI dysfunction is 80%. And I asked, well, do you know why that is? It's your question. Not clear. So it does obviously have something to do with permissiveness to certain microbes, but also we should all keep in mind that autistic individuals have very restricted diets. So their food consumption is not quite typical. They absolutely will not eat some foods. But whatever is the reason, the GI dysfunction is quite common.
Is there an age window where the dysbiosis has its effect?
That's
Because
another really
autism starts early, right?
another super important question. In fact, when we talked to the FDA about the study we want to do, of course, you would like to treat the youngest kids in an effort to do the most good. Because we don't have any safety data at the moment in a pediatric population. What we have to do is begin with young adults, then go to adolescents, and then go to kids, which makes all the sense in the world. But we will, as part of doing the study, actually hopefully learn something about that. But there is a notion that you might actually improve condition even in some of the older individuals.
C. diff infections are associated with depression.
Yes.
Has anybody tried your compound or thought about your compound in the context of depression?
No, but that notion has been suggested. So there are actually quite a few other conditions that could be considered. Depression is one, the Parkinson's is another one, multiple sclerosis is another one, which actually is tied with Clostridium perfringens. But we would like to begin with one that for which we both have the means and makes the most sense, and then consider others as we go along. But that is an idea that, in fact, could make sense.
Thank you so much. That was wonderful, very enlightening. The studies that you spoke about were for children. Could you speak to autism in adults and how this might possibly roll over into working with adults?
Yeah. I mean, the answer is I don't know. But that is a really important question. What I do know is that GI symptoms very much are present in adults as well. What is causing them, and to what extent you might undo some of the damage that had been done over a longer period of time, is an open question, and we just don't know, but we would very much like to get to it. So again, the best case is you do the most good, if we can show efficacy and safety in the very youngest population to extend the benefit, for as much of the time that the individual has. But for sure, if it works in adults, that would be just great, even to some extent. Yeah.
So, Nebojsa, I've seen roughly this information several times now. It never ceases to just amaze me, both in the downstream effects that you can show and in the cleverness of the target that you picked. And my question is really, it might be just the way you show us the phylogeny, but the MET-RS1 clade seems to be way overrepresented in nasty bugs, and the MET-RS2 clade has got everything else. Is there a reason for that? Is that just, are you showing us just the bad ones and the good ones, or is there some more basic thing going on here?
So if Norm were here, and I was hoping he would be, he would say, well, Larry, the reason that it seems that way is because we know way more about pathogenic bacteria that affect us than other bacteria. So, in a sense, that group is overrepresented by itself. So, I didn't show you a lot of the organisms that are known to have MET-RS as well, just to kind of simplify the slide. But that we should always keep in mind that bias, that we humans are biased, in terms of our interest in organisms that affect us.
you mentioned Parkinson's. There's a lot of overlap in the Parkinson's and autism microbiome, and I've been studying the Parkinson's microbiome for about 10 years.
What's the question, Wendy?
And
Oh, yeah.
one of the things that's interesting in our data set of Parkinson's samples that we collected is that the methionine salvage pathway is upregulated. So that would
That's
be
right.
an indicator, and they have perfringens and Boltier.
Yes.
And I tried to get somebody to look at that about 7 years ago, and I just couldn't get anywhere.
I'm really happy you brought this up. That is absolutely the case, and I would love to understand that better.
Well, I'll find you after, and you can look at my data.
Tom. Oh, you need a mic. You need a mic.
All right, I'm slipping in the
Oh,
bush.
sorry, Jason.
Nice talk.
Thank you.
I suspect most of us here agree that the RFK's autism hypothesis is shitty, and I just want to say your shit hypothesis is better than his.
Yes.
But, we talked about this at breakfast a while ago, and sort of the first thought that came to mind when I saw the P. Creasol stuff is, is there a microdose where you can turn on autism and turn it off? And, a year ago I might have said turn on your inner Elon Musk for a week. We won't say that now.
Yeah.
But was it fully reversible, and did they run a lot of cycles? And could you imagine you want to think really hard about something and you microdose this? And
Yeah, right. I mean, that's not a crazy idea. I think it would be difficult to do for sure in humans. And, with the RFK stuff, yeah, for sure, a lot of the ideas are completely misguided, especially as they relate to vaccines. I think we all have a tendency, when you think about a person like RFK, to say, well, this person is wrong about everything. I mean, it's an intrinsic human nature thing. The notion that we ought to spend more time thinking about environmental causes of autism, I don't disagree with that. Because if you think at the rate of increase of autism over the last 20 years, that's not genetics. And it's also not only about better diagnosis. In fact, if you look only at the most severe cases, those have been increasing as well. And it's really not clear why. So I think it would be good for us to think about why that would be in an open-ended way. And so, I mean, the pair crystal data are so amazing that it does make you think, well, let's use it in some way.
One last.
Nebojsa, have you considered looking at people with Down syndrome? They have a pretty dramatic increase in autism and in intestinal issues.
Yeah, I mean, I do happen to know, Tom, that you know way more about Down syndrome than I do. I mean that could make sense. It is a clear genetic condition, and it's not a subtle one. An entire chromosome. There's an entire extra chromosome, 21. So maybe that could make sense. But I don't know that much about it. What I don't know at all is about the composition of the microbiome and whether the spectrum of CRS3 matches in any way with whatever dysbiosis might be there.
that's why I asked.
Yeah, I don't know. I don't know. Possibly. I mean, it does appear to be a contributor. Again, I just learned this 2 weeks ago. That effect size is not huge, but it couldn't be good. So I don't know to what extent it can explain this exponential rise in autism cases.
With that, thank you so much. Thank you. That was incredible. We can find all the speakers in the break.